everyone to another episode of Nerd RX podcast, and I'm your host Barkha. Today we have Dr. Chavali who is going to talk about isothermal titration calorimetry, which I have absolutely no idea about, so I'm going to learn with all of you. Welcome, Dr. Shashank, to the show.
Thank you so much, Bharka. Thanks for having me. It's this is the first time I'm doing, and I'm really looking forward to this. Thank you so much for inviting me.
You are most welcome. I'm so glad you took some time out of your schedule to be here with all of us.
It's
So
my pleasure. Yeah.
before we jump into the topic, we would
hmm
love to know about your education background and how did you get to this point in your career?
Okay, sure. So I started off my scientific career as an undergrad researcher. I've actually during my 11th and 12th grade, that's when I decided to pursue science. And I really somehow felt that uh, discovering something that no one has discovered ever, or like seeing something that no one has seen before for the first time, was something that really clicked well with me, and I really wanted. I started became becoming interested in research for that reason. So I I wanted to. I didn't know what I should research.
Yeah.
So the most the most cool thing at that time was research in life science, because like I used to hear terms like DNA and cloning and like all that. It sounded very cool to me.
Mhm.
But I had no clue, because like I had mathematics and like physics chemistry background up until 12th standard, and biological sciences required some background in biology.
Biology.
uh,
Yeah.
right. So that is something I thought maybe I would develop. I mean, I had like high school biology. It wouldn't be that bad. But so I was sort of an outlier when I decided to study biology, or like biological sciences. In especially in India, where I grew up. I'm from Hyderabad in India. Engineering is like is the thing. Like everyone are supposed everyone like that's the thing that
That's
everyone's
the
supposed to do.
Asian parent thing. It's either
Exactly.
engineering or medicine.
Exactly. So I was also like one of them. I was thankfully, like I wasn't like pressurized by my parents or like anyone around me to like do engineering, but I was also like I was confused. Like I wanted to do research. I didn't know if I was supposed to do engineering in like biotechnology or like biomedical engineering, or whether I'm supposed to, like do a BS, Bachelor of Science, in like chemistry or biology or stuff like that. But I ended up doing engineering. I studied biotechnology in Yale University in India. It's in uh, it's in a place called Vijayawada in Andhra Pradesh. So, so the I mean from the time I joined, I was like super interested in like jumping into research projects, and I've heard at least like when I was applying to different colleges, that this particular university had a very good biotechnology program, and the faculty there had pretty good doctoral and postdoctoral experiences. So, which is true. So when I joined there, I started. I didn't know what project to do, but like I slowly, once I got into my uh, sophomore year, I started like appreciating the science behind
Yeah.
drug discovery, the science behind cancer biology, the science behind genetic engineering, and so that's when I slowly like started into like mini projects, and I collaborated with one of the faculty members there with the structure biology project. Um, and that's where my first like legitimate research experience started. And for some reason, I really felt like looking at things at an atomic level, like looking at
Mm-hmm.
structures of proteins, looking at atoms. Like it felt really cool, because if we are able to connect a function, uh, a phenotype or some sort of function back to an atomic level, I think that is, in my opinion, a very comprehensive understanding about a particular biological function.
Correct.
So, I became very interested in like knowing the chemistry of interactions, knowing the physics of interactions, so in my first project, that's how I started into structure biology. And so after I finished my bachelor's, I was able to I was a co-author on a paper that came out like at the time I was graduating, and I was like really happy with that, because that is something. So I did not, study like at a top university like an IIT or something in India, and to be able to get something
Recognition.
out
Yeah.
that anyone in the world can read. It can be cited by people. That felt like a pretty good achievement for me.
Yeah.
So from there, then I went on to do my master's at Rutgers University in New Jersey, where I slowly got into computational biology at that point, because so my research work as an undergrad dealt with computational biology. I did not do any experiments there. Like I used to do some molecular docking studies and stuff like that, and that uh, like really uh, uh, like made me focus on these molecular interactions, which, because like they can be seen on a computer and they can be simulated on a computer, I really found it very cool. So I did my master's in computational biology in Rutgers, and there I learned something called as molecular dynamic simulations. And
Mm-hmm.
this basically is to simulate proteins or like nucleic acids or lipids under physical forces. Like they're like basically you're just taking a tiny little part of a cell and putting it on the computer and simulating it and see what it does. And that idea itself was like so fascinating to me, because you're not just understanding the biology, but you're also understanding the physics. There
Yeah.
you have to understand the physics, because if you're simulating something, then there has to be all sorts of physical forces that are applied or that are applicable in a cell on the computer. So that really improved my understanding about biophysics, because that's the first time I appreciated the import. I knew the term biophysics before that, but I actually appreciated how important biophysics is in order to understand biological functions is when I started learning more about molecular dynamic simulations. And
Mm-hmm.
um, yeah, so there I used to, so I worked on membrane proteins, and simulation of membrane proteins, and understanding how how metabolites pass through membrane channels. And all of that made me, so up until then I was interested in a particular technique, or I was interested in a particular field, like structure biology or computational biology or biophysics. But after that, I became interested uh, in like finding a question to answer.
Mm-hmm.
Like I didn't care if it was if it was a question that only computational biology can answer, or if it is a question of molecular biology. or if it is question of biochemistry or like structure biology. I didn't care about that. I wanted to uh, come up with a question, think about how I would approach it,
Yeah.
and go from there. So then I joined University of Rochester in upstate New York into the biophysics program. And obviously, during the first year, I rotated in several labs, and I started appreciating uh, the research on RNA. Up until then, like again, like just how my transition from bachelor's to master's was, my transition to master's, from master's to uh, my PhD program was also similar, where I didn't know much about RNA research.
Yeah.
And so when I started learning about how RNA also like physically behaves like proteins, it also falls into structures, very complex complementary structures. And I was like completely blown away by how uh, I didn't know about this. Like we
Yeah.
all study about proteins, we all study about secondary, tertiary, quaternary structures of proteins, and how enzyme inhibitors need to be designed, and like or how GPCRs are to be targeted. But targeting RNA structures really is an underexplored
Yeah.
field. And University of Rochester, and if any listeners are interested in RNA research, University of Rochester is one of the best places in the US that does
Wow.
RNA research from any perspective, like be it computational, cell biology, biochemistry, pharmacological. There's like every sort of
Hmm.
uh, approach into
Yeah.
studying RNA. So yeah, so there I started learning more about RNA biology, RNA structure biology, RNA biophysics, and I joined a lab which developed therapeutics that target RNA. So it's a structure biology lab. My basic interest has always been, like I said, studying things at an atomic level. So I was studying RNA at an atomic level there. Like I did a lot of X-ray crystallography and like uh, isothermal titration calorimetry that I'm going to talk about.
Hmm.
Other binding experiments such as surface plasmon resonance, and I also did like a structural bioinformatics project during COVID because like we were not allowed to go into the labs.
Yeah.
And somehow this project came up, and I started working on that, and that led to a very interesting finding, which was published in JACS, Journal of American Chemical Society.
Wow.
So yeah, so uh, I basically try to understand how to target RNA structures.
Hmm.
Uh, my advisor, Dr. Joe Edicond, is an expert in RNA structure biology and RNA biophysics, and he like taught me about how to approach uh, 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 designing drugs against RNA, and what sort of things to keep in mind uh, while optimizing things. And uh, so I learned a lot in those 5 years, where I started off as a person who knew nothing about RNA biology, but at the end I came out with a lot of knowledge in RNA structures and RNA biophysics. And the specific RNA that I was working on was a non-coding RNA, which is part of the HIV genome.
Hmm.
So yeah, so I'll talk more about that when I'm explaining
Right.
ITC. But yeah, so from then, I then the most favorable transition from X-ray crystallography for any structure biologist is cryoelectron microscopy.
Yeah.
So in 2007, cryoelectron microscopy won the Nobel Prize, and that's when I mean, it was very well known in the structure biology field, but that's when people outside of the structure biology field started appreciating that.
Yeah.
So I started. I was also one of them. I really uh, uh, thought cryoelectron microscope is something that uh, it's a new thing, relatively new thing, but it's still uh, there are so many things that can be done with it, and that's still underexplored.
Yeah.
That and cryoelectron tomography, these are two things that are immensely contributing in the scientific community to come up with drugs. Like even in the case of COVID, there's so many structures that came out, and cryo-EM pipeline is so well optimized at this point that like people with good resources and money can crank up structures like every month.
Yeah.
So it's really impacting the scientific community like across different disciplines.
Hmm.
I felt so when I was transitioning into a postdoc. So now I'm currently a postdoctoral associate in Yale University, where I'm working on understanding the molecular mechanisms of cytoskeletal and motor proteins. So I found a gap in the field where, like in terms of muscle related diseases and muscular problems, there is the biophysical and structural aspects of understanding uh, these diseases was like very it was underexplored. So so that's when I thought maybe I should learn more of cryo-EM and uh, like learn more about how things move inside the cell, like as in how motility, cellular motility happens, and what happens when like molecular motors bind to like cytoskeletal elements. How are they regulating a diverse set of functions, and how these can be applied in understanding muscular problems and any muscle related diseases, because that is like especially there are so many genetic diseases in the that are neuromuscular or musculoskeletal
Hmm.
genetic disorders, and the focus has always been to come up with gene therapies and come up with like other therapies that are not really structure biology based, but they're all great. I mean, there's like a lot of important research going on there, but I think structure biology can also aid this and like help or bolster this whole effort of treating muscular problems. So I think I felt that that was the gap that I think structure biology can fill. So I wanted
Yeah.
to learn uh, because I had like a very strong background up until now in like crystallography, molecular dynamic simulations, and like other computational techniques, which always all of them dealt with structures. So this is the only thing that I was not like super well exposed to cryo-EM. So that's when I decided to learn more about that, and yeah, I joined Yale last year in July, and I'm still going.
Yeah. Well, in the beginning, you mentioned you did your
Hmm.
bachelor's from a very small college, but look at you now, you're at
Hmm.
Yale, one of the best.
Right, right. Yeah, that is when I do look back, I do feel very happy about that, and
Yeah.
like, and I think like if I can come across from that point to this point, then I think like anyone can do it.
Yeah.
So I would like suggest like uh, to all the listeners that like if you're passionate about something, don't give up.
Absolutely.
Yeah.
Well, thank you so much for taking over this journey of how you got here. And it's honestly quite inspiring. Like having a paper out of your bachelor's is a big deal. So, congratulations
Thank
on
you so
all
much.
your achievements.
Thank you so much.
Yeah. So, let's get into the topic now, and
Sure.
we
Yeah.
will talk about ITC. So,
hmm
Shashank, what is
hmm
ITC and what about it got you interested in it?
All right. So ITC, like you explained in the starting, stands for isothermal titration calorimetry, and so in any sort of interaction to happen between any biomolecule, that could be between a protein or RNA, or protein and a metabolite, or RNA or a metabolite, anything. It has to that interaction is has to obey certain laws of physics.
Mhm,
So thermodynamics is one thing that is extremely important that to govern certain interactions, the favorability of a particular interaction, an enthalpic or an entropic contribution towards an interaction is extremely important to study, because if we understand how two biomolecules are interacting, when that thing is messed up, or when there is a mutation, or when there is something that's not working in the cellular environment, we know how to target them, we know how to repair them. So so uh, so when you zoom into a cell, when you zoom into the in the cytoplasm, when you look at two particular interactions between two molecules, then uh, they should obey the laws of thermodynamics. So, in order to come up with drugs that can mimic those interactions, in order to come up with drugs that can stop those interactions, there are so many possibilities of targeting these interactions. And so when I worked as a doctoral student, when I was trying to design therapeutics that bind to RNA, my goal was to come up with cyclic peptides that target RNA. And
mhm,
because these peptides are a small chain of amino acids, right?
mhm,
So when I wanted to design these molecules, I had to understand how these two molecules interact, and in order to optimize these interactions, in order to improve their affinity, in order to increase the specificity towards a particular target, we need to understand how they both interact, and understanding the thermodynamics is one of the best ways uh, to to basically like figure out how to approach, how to target this particular RNA or protein or enzyme or whatever. So that's the reason I became interested uh, in learning ITC. Uh, I wanted to see how two biomolecules, in my case it's RNA and peptide. I wanted to see how they interacted, with what affinity they interact, what is the entropic and the enthalpic contributions towards these interactions. And in order to improve this binding, in order to improve the specificity, how should I engineer it? How should I optimize it? So these are the questions I wanted to answer, and ITC is the best way to do that.
Well, okay. So, when you are designing an experiment for ITC,
hmm
what are
hmm
the steps involved and how long it typically takes from start to finish? Could you go over the steps involved quickly?
Sure, sure. So one thing that is required in ITC is you basically need purified samples, because, like I said, you're looking at interactions between two distinct biomolecules. We need them to be super pure. So if I'm working with RNA, I need to purify a particular sequence of RNA, uh, and if I'm working with protein, I have to purify a certain protein that is it has to be extremely pure. If there are any
Mm-hmm.
contaminations in that, then we are measuring interactions with other things.
Okay.
So we to make sure we are basically uh, seeing the interaction between two things, we need to make sure those are the only two things we are putting
Mm-hmm.
into that instrument. So that is the first thing. Which so if the experiment itself doesn't take too long, but coming to that point
Yeah.
takes too long. Like I'm sure you or like the listeners would appreciate how difficult protein purification is,
Mm-hmm.
and RNA purification is another difficult step, because one thing about RNA purification is there are like just like proteases, there are RNases everywhere. Like even as we speak, we are like spitting out RNases, and so we have to be extremely careful while purifying RNA samples, uh, and that takes a while. But after we come up with those, I also worked on purifying proteins. I worked on looking at the interaction between protein and RNA. But basically, coming up to that point is the most time-taking step. But after getting to there, as an example, like I worked on RNA and protein interactions. So from there, the using the instrument is actually not super difficult. It's pretty simple given we understand the basics of how the instrument functions, which we'll go through
Mm-hmm.
in a minute. But uh, so the so yeah, so the time that usually is required to complete this experiment is probably, I would say, 3 to 4 hours. And now
Okay.
the instruments have gotten smarter. So like some experiments like take like an hour, or
Mm-hmm.
yeah, I would say an hour, with the most state of the art instruments. I'd say it would take about an hour. And so once I have the purified protein and whatever biomolecule, then the instrument has two cells as in like two chambers basically. One chamber is filled with water, just water, distilled water, and the other chamber is where we have our sample. So one of the samples, whether it can be a protein or RNA or any biomolecule. Usually, it is the if you're looking at the interaction with between a protein and a small molecule, then the small molecule, the protein will be in the chamber, and there's a syringe in which you load your small molecule. So in my case, the RNA is in the chamber, and the peptide or protein, the one that should go and interact. The target usually is in the chamber, and the one
Mm-hmm.
that needs to go in there is usually in a syringe. And that syringe, so once we load the RNA in the chamber and the protein in the syringe, we basically start the experiment. So what the instrument does is it releases like extremely small amounts, like nanoliters of the sample from the syringe into this chamber, and it mixes the syringe mixes the liquid or the solution in the chamber. So if an interaction happens, if A binds to B, and if
Mm-hmm.
that is an exothermic reaction, which means if it's releasing heat, then the instrument senses that heat, and it utilizes power to increase or decrease the temperature of the reference cell, which has water. So uh, like I said, there are two chambers. One chamber has water in it, the other chamber has our sample in it. So if there is an interaction that happens inside our sample chamber, then it will either release an heat, or it will absorb heat.
Correct.
If it's an endothermic, it absorbs heat. So once it absorbs heat, or once it releases heat, the temperature changes in the chamber. So once that temperature is changed, the sensors recognize that, and there is a feedback into the machine that increases the power to heat or like cool the reference chamber.
Mhm.
And once that is done, uh, this power is basically integrated over time to generate a heat. So we basically get from the experiment, from the first injection, we get like a heat value.
Mhm.
And now this process goes on in an iteration. So basically, once a small nanoliter amount is injected into the chamber, you're going to inject the same amount again. Now
Okay.
if there are like 100 copies of your protein or RNA there, and if like in your first injection, like 10 copies of them have been occupied by the small molecule or your drug or whatever, then there are like few more left, right?
Yeah.
So in this iterative process, you keep injecting sample, and you keep measuring heats. You keep increasing the machine does this, but the machine increases the power to balance the temperatures in the reference chamber and the sample chamber. And slowly the once like all of the protein molecules or all of the RNA or whatever biomolecules are saturated with the ligand that you're
Mhm.
injecting, then there is a plateau that is reached. So basically you're going to get a curve
Mhm.
that is called an isotherm. And the heats keep reducing as we go towards saturation, because like there are fewer copies that need to be occupied after each injection, so
Mhm.
fewer copies of biomolecules. So this basically the machine basically measures the power required to balance both the temperatures, but that power over if it's integrated over time gives you the uh, heat, the energy uh, that is calculated, and the slope of this curve gives the binding affinity of that ligand to the molecule, to the macromolecule. So this basically gives us two things. One is the binding affinity, and one
Mhm.
is the heats of binding. So once we know more these two things, we can use laws of thermodynamics to calculate the whole the free energy of binding, which is delta G, and we already know delta H, and the only thing we need to know is T delta S. So I just want to remind the formula that we all usually use for uh, uh, the Gibbs free formula, which
Mhm.
is the delta G equals delta H minus T delta S.
Mhm.
And so T delta S stands for entropy, delta H
Okay.
stands for enthalpy, and
Mhm.
delta G is the Gibbs free energy.
Gives free energy.
And
Yeah.
the the so the once we calculate these, the lower the delta G is, the more favorable the interaction is.
Okay.
So if we have two interactions with one delta G is let's say is like minus 10 kilocalorie per mole, and then there is another interaction that is minus 5 kilocalorie per mole. So the first interaction is more favorable than the second one,
Mhm.
and that so that is like the thermodynamic uh, understanding about so anything that is more favorable has less energy, like lower
Okay.
energy. So yeah, so or uh, think about it in a more intuitive way, like if something is favorable, we don't have to apply a lot of
A
energy.
lot of energy.
It
Correct.
happens effortlessly,
Yeah.
right? So it is a crude example, it's a crude analogy, but that's a basic thermodynamic understanding of how low energies are contribute to favorable interactions. So I can obviously like we don't have to get into the details of how uh, the uh, like we don't have to get into like thermodynamics here,
Yeah.
like hardcore thermodynamics, but
Yeah.
but so delta G actually, maybe I forgot to mention this, but delta G, we know of delta H from the experiment, but delta G is calculated using a formula. Delta G also equals negative RT log KD, or negative log K, and K here is the binding affinity.
Okay.
So once we get the binding affinity from the slope, we calculate the delta G, we calculate delta H. Now from that we calculate T delta S, and now magically we have the enthalpic contributions, which is
Yeah.
delta H, the entropic contributions, the T delta S, and the overall favorability of an interaction, which is the
Mm-hmm.
delta G. So so we don't have to do any of this. The software does all of this. We just need to inject the samples into the syringe and into
Mm-hmm.
the sample chamber, and all the magic is done by the instrument.
Wow, that was a very detailed and easy to follow explanation. Thank you so much for that. Like, I could picture the entire thing in my head, and I have no idea about
Awesome,
it.
awesome. Great,
Thank you
great.
so
I'm
much.
glad. Yeah.
Another thing I wanted to ask is, are there any
Hmm.
alternative techniques to ITC?
Right. So there are and there are not,
Mm-hmm.
as in, so one of the main reasons to perform ITC is two main reasons. One is to know the binding affinity of a ligand to a molecule, macromolecule, and the other is to know the thermodynamics. But usually, knowing the thermodynamics is not always necessary. Like it is necessary to optimize drugs to know, uh, uh, like, I mean, it depends on the question, basically. It
Question. Yeah.
depends on what, if you're developing a drug and you want to, if you have like tons of drugs that you want to screen, ITC is not a good method at that point, because you need
Mm-hmm.
a lot of amounts of your protein and drug, and you have to do it one after the other. There are more high throughput ways to, filter certain compounds out, and yeah. So at that scenario, ITC is not the best experiment to do. But if you have like a few hits and you want to optimize those hits, you want to increase the binding affinity, you want to change the functional groups of a drug molecule, you want to change the protein sequence, you want to change the peptide sequence, uh, that we know. And the most useful approach of ITC is how we can relate structure to function.
Mm-hmm.
So we are basically, if we look at a crystal structure or a cryo-EM structure and we see two amino acids interacting, and if we mutate that amino acid and we perform ITC, we will see that interaction lost in terms of how much delta G is lost. So if one
Right.
hydrogen bond is lost between one amino acid, between two interactions, then we would get a delta G of negative 1.5 kcal per mole, which is the energy required to form one hydrogen bond.
Mm-hmm.
So that is the level of detail we can understand. So I mean, it's a bit of a digression, but the alternate to do this is we can perform other binding experiments, which is like surface plasmon resonance. You can perform fluorescence assays to
Mm-hmm.
get the KD. Once we get the KD, you can also, like biophysics people uh, will not like this, but you can, in theory, calculate delta G from KD, because all you need to do is plug in the KD value into
Mm-hmm.
a formula, delta G equals negative RT log KD. But the biggest problem with that is KD has units, right? Like binding affinity has micromolar or millimolar or nanomolar or whatever units,
Mm-hmm.
and you cannot take a logarithm of something that has units.
Yeah.
So the biggest problem, so it's not delta G anymore. It's something called as delta G naught, which means delta G at steady state. And steady
Okay.
state assumes 1 molar of the solution. So you're basically in that logarithm, you're you have a denominator of 1 molar, which is the
Mm-hmm.
concentration. So the concentration values units gets cancelled, and now you can take a logarithm.
Mm-hmm.
But you will not get enthalpic and entropic contributions from other binding techniques. And sometimes you don't need them, like I said.
Yeah.
If you want to filter out a lot of compounds, if you have 100 compounds and you want to filter and come to like 10, then ITC is not a good way to do that. The more they're more like very effective high throughput fluorescence assays and lots of other assays that you can do to like filter out the stuff that are not binding to your target. And once you have a few, then you can start performing those. And step by step, like it's like a Lego piece. You're constructing something, and
Yeah.
if something doesn't fit there, you're taking that out. You're putting another thing, and you're specifically designing something to bind to that pocket. Because in ITC experiments now, here you're only looking at two things. You're not looking at anything that is complicated. So
Mm-hmm.
that actually increases chances of coming up with something that is very specific.
Mm-hmm.
So yeah, so there are alternatives. There are actually very good alternatives to ITC, but those alternatives don't ask the same question.
Yeah.
Sometimes those are important, and sometimes ITC is important. So yeah,
Yeah, it depends on the question you are like the answers you're looking
exactly,
for, like your experiment design.
exactly. Exactly,
Okay.
exactly. Yeah.
So is this technique user friendly or is there like a big learning curve to it?
Yeah. So the technique itself is user friendly, but the most challenging part that is kind of underappreciated is how these results can be interpreted.
Mm-hmm.
So, so usually, if one compound binds to one protein molecule, the binding stoichiometry is like 1:1. That
Mm-hmm.
is simple. That is something ITC has figured out. So that is a simple model. So if you get data points, it is easy to come up with a curve that fits all the data points.
Yeah.
If the binding is 1:1. But when the binding gets complicated, that is, if it's like cooperative binding, or if there is sequential binding, or if there are two ligands binding to uh, one protein molecule on two different sites, or so that is when the data looks different. It's not that straightforward, and
Mm-hmm.
interpreting that data, coming up with actually like people develop algorithms like statistical algorithms to fit these complicated data points. And because everything that we understand, that we take out from an ITC experiment, the most important thing is binding affinity and thermodynamics. And this is heavily dependent on how good your fit of the data is. And if your fit is like noisy, if it doesn't cover all the points, then your value is not believable.
Mm-hmm.
So you're then that is the thing that is very challenging for someone to understand. So the technique itself is very easy to master. You can like once you purify your samples, like injecting sample, you have to be a little careful about using the instrumentation, which is the case with any other like biological
Yeah.
experiment. So that is all you need. But once you like get the results, making sense out of it, and coming up with good fits, understanding, believing those fits, like doing multiple replicates to improve your noisy data, all of this is time consuming, and it's a difficult thing to master. But it's achievable. But if your system is complicated, if your binding is complicated, if something fishy is happening inside of that chamber between two molecules, if it's coming on and off, so there are so many things like that, and that is when it becomes a little tricky. But if if it is simple, if the binding is simple, you want to just you know that something binds 1:1, you just want to optimize it. Then you do the experiment, you get a decent looking curve, the software fits it for you, you will get a KD, you will get a delta G, you will solve everything. That is great. So but that's not the case usually,
Yeah.
because yeah, some things are complicated. But yeah, so yeah, to answer your question, it is user friendly, but it can also be challenging when you're analyzing the data.
Yeah, if science was easy, I think everyone would do it.
Exactly, exactly. I know, absolutely, yeah.
I think we spend more time in troubleshooting than doing the actual experiment is
Than doing the actual experiment, and that's how we all learn. That's
yeah,
how you're absolutely right. If everything works magically and you have a 1:1 stoichiometry and like everything is great, there's nothing you've learned
exactly.
other than like loading things onto the machine, that which
Hmm.
anyone can do once you show them how to do. So, so the actual crux of the science, or actual crux of the problem, comes when you're analyzing that data
Hmm.
and you're inferring the binding thermodynamics, and oh, what functional group is doing binding there, what maybe there are two binding sites, maybe these two binding sites are cooperative, and these are so I mean, I can like point you towards so much literature that does a
Yeah.
lot of cool stuff, um, and in coming up with these data fitting algorithms. And yeah, so like you said, it's the troubleshooting that teaches us more than like the doing that experiment.
Yeah, absolutely. But that is fun too.
Hmm. Right. That is fun. Sometimes frustrating, but it is fun.
Yeah, it is very frustrating, but when you are doing troubleshooting and
Work it
finally
out. Yeah.
you work it
Yeah.
out,
Yeah. Yeah.
that is, I
Yeah.
think, a very
That sense
sweet
of
moment.
accomplishment is
Yes.
great. I
Yeah.
yeah, we have all been there. Yeah.
Yeah,
Yeah.
I've been working on one of my experiment from, I would say, last 3 years, and very
Wow.
it's called in vivo calcium imaging. It's not kind of,
Oh.
it is very tricky, where you
Oh.
image
I have
in
done
a,
in vitro calcium imaging
oh no, this is
in
in
cells.
vivo, and it's quite a unique technique where you inject and it's live, so you see the neurons in a live animal moving around in the cage.
That is
S-
so cool. That is so cool.
yeah, so that took me like 3 years, and I finally optimized it like last month. And when I got it, I literally screamed in the lab. Oh my god, I'll log with
Awesome.
my PI.
Congratulations on that.
Thank you so
Congratulations.
much.
Yeah, it's a great achievement. Yeah,
Yeah,
I can like so I did it in one of the rotations, and I thought in vitro itself was so difficult, and I
yeah.
can totally imagine how in vivo imaging can like, yeah, yeah, you should be proud of that.
Yeah, it's pretty fun,
Uh,
but yeah, I think the more challenges, the sweeter the victory feels, you know.
right, right, absolutely.
Well, so getting back to this,
Mm-hmm.
what would you say are some of the advantages and disadvantages? I know you covered few of them, but
Right,
just
right,
to
right,
give you an
right.
overview.
Sure, sure, yeah. Yeah, so the advantages are, you will know uh, basically the thermodynamics of uh, uh, binding between two biological macromolecules, or even like small molecules. Uh, the like I said, the biggest advantage here is how we can relate these results into structures. Now we have crystal structures, we have cryo-EM structures, the protein data bank, which is like a database of all structures
Yeah.
that have been solved till date. Like you can like randomly take one structure out. You can like purify that protein. You can mutate that protein, and if that uh, to eliminate a particular interaction, and you can see that interaction gone in an ITC experiment. And
Mm.
I mean, see as in not visually, but like
Yeah.
mathematically, you can see that thing is gone. So that I think is the most powerful application of ITC, because
Mm-hmm.
if you understand that, then and if you have a complementary structure, then you're, like I said, you're constructing a drug, you're like engineering a drug, you're engineering something that is so unique to that particular target that
Right.
like it has to work.
Mm-hmm. Yeah.
And so that I think is the uh, best use of ITC, is how we can relate structure and thermodynamics at the same time with one experiment. So that and sometimes we don't even need the structure. If we have
Mm-hmm.
like a starting structure of something, and okay, we have a starting structure of a protein, but we don't have a drug. So if we have narrowed down to like a list of 10 drugs, now we are like uh, basically docking experimentally one drug after the other. In my case, actually, it helped me so much. So I have the RNA, and I'm targeting it with a peptide, right? So when I target, so when I use the first peptide, I got like a binding affinity. I got like some enthalpic contribution, and we sat down and we thought, okay, we can still increase the enthalpy. We can
Mm-hmm.
still increase the favorability of binding. So we first engineered the way the peptide is linked. So it's a cyclic peptide. It's not a linear peptide. It has to be linked. So it was linked through a disulfide bond first. So where two ends of the peptide have
Okay.
cysteine residues,
Yeah.
and these two are cyclized. So that produced some result, but we slowly optimized that cycling strategy, and so we collaborated with a cyclic peptides expert. His name is Dr. Rudy Fasan. He was in the chemistry department at the University of Rochester, who has this extremely great sense of chemical intuition. Uh, so, so they basically engineered these linkers where we took out the disulfide, we added a methyl group, then we added an ethyl group, then a butyl group, then like a xylene group. Like we basically increased the length of the carbons that link these peptides, and we see improvement in binding. We see improvement in the enthalpic contributions. And
Mm-hmm.
then we have, okay, now we have the peptide with a good linker that connects, that cyclizes it. Now we changed the amino acids slightly. So because RNA is like negatively charged,
Mm-hmm.
we have like cyclic peptides that are full of positively charged residues, so that they can like bind to each other
Right.
electrostatically. But like you don't want it to bind to every RNA in the body,
Yeah.
right? Like if
Mm-hmm.
you if you do that, like you're basically it's a catastrophe. So so we engineered it in such a way that we added one arginine or one positively charged residue at each place, and we saw how it affects the binding. We did some other, controls, so that we had some commonly seen RNA inside the body that we tried to bind our drug with, and that didn't bind to that. So we tested the specificity. All of this is in theory called structure activity relationship.
Yeah.
So basically you have a structure, you have the activity of your compound, and you're optimizing it with the help of ITC and like
Yeah.
the structure.
Mm-hmm.
So sometimes you don't even need the structure. Like you can have like a protein or an RNA, and you can have your drug, and you can slowly optimize it. And once like you have one drug that is binding well, and it has great enthalpic entropic contributions and everything, now you can try to solve the structure of the drug along with your RNA or protein, and you will see those interactions there.
Mm-hmm.
So so that is an extremely complementary way, and like it is, to me at least, it's like fascinating how you can
Mm-hmm.
optimize things that is like so translatable
Yeah.
into coming up with drugs.
Mm-hmm.
Yeah, so this is one of the biggest advantage, and the disadvantage is, like I said, the binding becomes complicated.
Yeah.
The fitting is the analysis of data, and the fitting is a very big problem. And so sometimes you have to write your own like fitting code, like
Mm-hmm.
statistical. You need to come up with your own algorithm to fit that data,
Mm-hmm.
and I've seen that. So one of my colleagues, when I was a doctoral student, had this very cool project where he had like an RNA molecule that has two ligands that are stacked over each other that bind in one pocket, and
Wow.
so there were like two dips in the curve that
Mm-hmm.
he saw in the ITC experiments, and it's difficult to fit with all the statistical algorithms we have. It's difficult to fit some random data,
Mm-hmm.
and even if we fit, we don't know if it can be believed. So we had to collaborate with a computational group
Yeah.
to come up with a particular algorithm to fit that data that is believable. So that is one big disadvantage. If the binding becomes complicated, it's difficult to analyze. And the other disadvantage is you need like a lot of amounts of your biomolecules. Like
Yeah.
you need to purify a lot of protein. You
Mm-hmm.
need to purify a lot of RNA. You need a lot of compound small molecules or peptides or whatever. You need like large amounts of it.
Mm-hmm.
So that sometimes is a rate limiting step,
Yeah.
because you spend like weeks in purifying protein, and
Mm-hmm.
you may like, especially for someone, if they're starting out in this, and they may make some mistake, and
Yeah.
you use up like 50% of your protein in one experiment, and you have to do at least three experiments, right, to make sure, like, to replicate so that
Yeah.
like your answer is more believable.
Mm-hmm.
So yeah, so then it becomes a little difficult, but these are the two main disadvantages.
Okay, so let's talk about the cost involved here. Is this
Right.
instrument and even the consumables like expensive, or
hmm
any regular lab can set this up?
Yeah, so it is slightly on the expensive side.
Mhm.
It's not as expensive as an electron microscope, which is like $1 million or
Yeah.
something. But it is slightly on the expensive side, and usually, like I've seen that labs, uh, like like a department has one ITC so that like people can use it. Not
Hmm.
everybody wants to use it every day,
Yeah.
so like it can be a departmental thing where like
Hmm.
certain labs can buy it together. Sometimes, like, so in our lab we used it extensively. So in Rochester, when RPI got a grant, and he so we had our own ITC machine.
Okay.
I would say it is on the expensive side, but if it is something that someone wants to use, and if they see a lot of application for it, then it's a great investment.
Okay.
Yeah,
So let's talk about fun facts. Does ITC have any fun facts?
right, right. So it does, and I think it's pretty cool. Like AIDS was such a big, I mean, it is still a big problem. There is no functional cure for AIDS, but uh, like it was much worse back in like the 1990s or even
Yep.
early 2000s, where there was no antiretroviral therapy. So the um, antiretroviral therapy is like a combination of drugs that basically target different points in the HIV life cycle.
Hmm.
So all of the drugs that were developed to target these different points in the HIV life cycle were like targeting the HIV protease, for example.
Hmm.
The drugs that target the HIV protease were actually developed with the help of ITC.
Wow.
Like they were optimized. Yeah, so they narrowed down from like a bunch of drugs to a few, and once they had a good hit, they like functionally improved the binding of the drug, binding thermodynamics. They improved the enthalpic and entropic contributions, and so that actually is helping a lot of people right now.
Yeah,
So we don't hear a lot of HIV/AIDS related deaths now
that's
than like
anymore.
how we used to hear
Yeah,
probably 2 decades ago.
yeah,
And antiretroviral therapy, even though obviously like it it does induce side effects for sure, but it still is like improving the quality of life for a lot
yeah,
of people. And this therapy wouldn't have been uh, as good without the efforts of ITC, and like without the contributions of this methodology. So that, according to me, is like super fun. Yeah.
yeah. Like I work in an HIV lab, and I had no idea about this. So thank you so much for
Right.
sharing.
Of course, of course. Yeah, yeah, yeah. My pleasure. Yeah, it's really yeah, and thanks for doing this so that like many people would now appreciate
Yeah.
uh, how ITC is involved in drug discovery, and
Hmm.
maybe they also want to use, if they have some molecules and they
Yeah.
want to optimize. They want yeah, yeah. It's you can collaborate with people if you're not
Yes.
an expert in that.
Hmm.
That's the purpose of science.
Absolutely. And I think that is the purpose of this
Right.
podcast, connecting people and getting to
Exactly,
know more
exactly. Yeah,
things.
exactly, exactly. Yeah, awesome.
Well, so my last question for you would be, could you suggest
Hmm.
any interesting articles or protocols regarding ITC so that I can link it down in the description for our listeners to look up?
Sure. So, okay, there are two papers I want to suggest,
Hmm.
both from our lab,
Hmm.
which I think covers like a good set of applications of ITC. One is my own paper. It's called Cyclic Peptides with a distinct arginine-fork motif recognize the HIV trans-activation response RNA in vitro and in cells.
Okay.
So this is basically this is published in Journal of Biological Chemistry (JBC),
Mhm.
and so this has the story of how we optimized peptides to bind RNA with the help of ITC results. So you will see like a ton of ITC thermograms in that paper. That
Okay.
is one paper, and the other paper is a more uh, challenging paper from the same lab, which I talked about briefly. It's called a small RNA that cooperatively senses two stacked metabolites in one pocket for gene control. This was published in Nature Communications, and this is the
Wow.
one that has a complicated mode of binding.
Hmm.
And in this paper, like you will see how the authors came up with way to fit these
Yeah.
complicated uh, data. So I think these two papers. I'm being selfish and promoting our own thing, but
No,
I
absolutely.
think it captures the essence of ITC pretty well.
Yeah. Yeah. Okay.
Yeah,
Well, with this, I am going to end today's episode. And thank you so much, Shashank, for being here and sharing everything about
absolutely,
ITC.
absolutely. Thank you so much for doing this, and I wish you all the very best. And I hope this continues for very long. And thank you for like integrating and uniting like scientists across the world. Yeah,
Well, thank you so much. And it has only been possible because of scientists like you who are willing to participate in it. So thank you so much.
of course. Yeah, thanks so much. Yeah,
And listeners, I will catch you next week on another episode of NerdRx podcast. And in meanwhile, if you have any suggestions for the topics that I should cover, or if you would like to join me on the podcast and share something interesting, please email me at barkha@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.